Hello my dear students, I am Dr. Vaishaly Bharambe, Professor of Anatomy, and today I am going to teach you how to draw the microscopic structure of salivary gland. Before this, I'll just give you a very brief of how we are going to do this. The salivary gland is made up of parenchyma and stroma. What is parenchyma? Parenchyma is the secretory and conducting element, that part which will produce the saliva and carry it. So who produces saliva? The acini, which can be serous, mucus or mucus with serous demilunes. And who carries the saliva? The conducting element, which is the ducts. So there can be intercalated ducts, striated ducts, interlobular ducts, etc. What is stroma then? Stroma is that supporting capsule. Okay, so connective tissue capsule, connective tissue within the lobes, between the lobes, okay, all this connective tissue becomes the stroma. So when we draw salivary gland, we draw each of these elements. Let's begin the process of drawing by first drawing a serous cell and a serous acinus. Come. So take your color pencils, the H&E pencils, and let's begin to draw a pyramid shaped serous cell. So draw one serous cell here, remember apically microvilli. Draw one more serous cell right next to it. Let's now stain the cells such that the cell is apically eosinophilic and basally basophilic. Draw a large rounded nucleus, a little basally placed. Color the nucleus. Let's now begin to add some zymogen granules to the apical region so that the apical region looks further eosinophilic. Add the Golgi complex, add the rough endoplasmic reticulum, add the microvilli, and there you have the typical serous cell. Such pyramid shaped cells will together create a serous acinus. So a rounded acinus with these pyramid shaped cells placed at the periphery so that the lumen in the center becomes quite narrow. Place the cells in this manner. Now let's stain the cells. Ensure that you stain them well. Rounded nucleus in the center. Draw the zymogen granules apically. As you can see, I have left a little space at the edge here. This space is for a special cell called the myoepithelial cell. Draw its flattened nucleus, stain the cell. Remember, this cell squeezes the serous acinus, helping it to release its secretions into the ducts. This is a serous acinus with a myoepithelial cell. Similarly, we will now draw a mucus cell and then a mucus acinus. Come. So each mucus cell is somewhat columnar in shape. In the apical region, you can see that it is showing secretions from the apex and some secretory vacuoles here, which are actually the mucinogen granules which are collected apically. Because there are so many mucinogen granules, the nucleus of this cell is flattened and quite basally placed. Let's color the nucleus. Let's draw the other cell organelles like serous cell. Let's now stain both the cells. You saw that these cells are very light staining, especially in the apical region. The mucinogen granules are lost during the staining section, so that portion looks even lighter. Let's use these columnar cells, arranging them around a large acinus, creating a mucus acinus. As you can see, the size and the shape of the acinus is quite longitudinal and large compared to the serous acinus. Arrange the columnar cells now along the periphery, creating a somewhat simple columnar epithelium. The lumen is large and tubular. Stain the cells light. If you look carefully, I am drawing fine mucinogen granules in the apical region now. Let's place the nuclei flattened and basally. The last thing now is to add the myoepithelial cell at the periphery. Color it, draw the nucleus. Remember, this will squeeze the mucous acinus, helping to release the secretion again into the ducts. So that's your mucous acinus with a myoepithelial cell. The third element we have to draw is the mucous acinus with a serous demilune. So draw a large acinus and draw with columnar epithelial cells. Now draw a crescent shape on the edge and draw pyramidal cells here. Let's color the mucous acinus first with a light color, apically even lighter with mucinogen granules. Let's color the serous acinus dark, draw large rounded nuclei and flatten nuclei at the base of the mucous acinus. As you can understand, this is a combination of a mucous acinus with a crescent serous demilune. It's a half moon shaped crescent shaped serous demilune. Color the nuclei and there you have a mucous acinus with a serous demilune. Here you should understand that the serous secretion passes through fine canaliculi and also releases into the lumen of the mucous acinus, thus mixing the secretion from both the types of cells. With these three acini, you have understood these three acini. We will now go on to understand the basic structure of a salivary gland. The salivary gland basically consists of an outer capsule which gives out septi, dividing the salivary gland into smaller lobes and lobules. The interlobular septa are dense connective tissue, while the intralobular connective tissue is loose connective tissue. Within this loose connective tissue, we will now add the ducts. So here is one duct, here is one more, and let's add one more within the septum. The intralobular duct is called intercalated duct. Sometimes it can have a slightly taller epithelium with a few striations at the base, in which case it is called as a striated duct. See how I am drawing a striation at the base. The interlobular duct may contain tall columnar to stratified columnar epithelium. Let's draw that as well. Remember, these are just elements. Next time you are going to mix up these elements to create the salivary glands. So that is the basic structure of your salivary gland. We will now draw first the mixed salivary gland, then the mucous, and then the serous salivary gland. Let's begin. Let's draw some rounded acini and some longitudinal acini, indicating that we are drawing serous and some mucous acini. Keep place for the serous demilunes and the myoepithelial cells. In this case, I have drawn pyramid shaped cells, indicating that this is a serous acinus. Some more pyramid shaped cells. Pyramid shaped cells in the serous demilunes. Now columnar cells in the mucous acini. Have patience, this takes time. That's a myoepithelial cell that I have added, and then I am drawing the mucous acinus with columnar cells. Some more pyramid shaped cells indicating a serous acinus. Let's now begin to stain the serous cells. Remember to stain them dark, eosinophilic. We can't keep drawing the basally basophilic, apically eosinophilic. It is difficult. So usually we stain them eosinophilic in such diagrams. I have colored the myoepithelial sites a little different so that they can show as separate cells. Draw the rounded nuclei in each of the serous cells and in the serous demilunes as well. Color them. Remember to draw the myoepithelial cell nucleus as flattened. Let's now begin to add flattened nuclei to the mucous acinus, basally placed flattened nuclei to each mucous acinus that you have drawn. This takes patience and time and meticulousness. Now I will add ducts to this. Draw the epithelium of ducts. This is the intercalated duct. Now let's support this with the help of loose connective tissue. Begin to color the tissue in between. Develop it so that it looks real. Stain it well. Let's now begin to add collagen and reticular fibers in between and a few capillaries with the nuclei of capillaries. Similarly, develop the other side. Now understand that you are looking at two lobules with the tissue in between being the interlobular septum. Okay, so now we have developed two lobules. Let's begin to work on the interlobular septum with the interlobular duct. So this will have tall columnar and at places stratified columnar epithelium. Draw it, color it, add the nuclei. Now let's add a few capillaries to this with the nuclei. This lower portion, I will add one more duct here. If you want, you can add striations to the basal region and call it a striated duct. Let's add connective tissue which is dense in the interlobular region. A few nuclei, and there you have the diagram of your mixed salivary gland. There is something I still would like to add. I am missing my zymogen granules. So let me now begin to add the zymogen granules to the apical region of the serous acini. And there you have the diagram of your mixed salivary gland. With this in place, it should not be difficult for you to develop the mucous salivary gland diagram and the serous salivary gland diagram. So for mucous salivary gland, you have to draw large acini, typically mucous acini, with a few ducts. Make place for serous demilunes and myoepitheliocytes. Now draw the simple columnar epithelium. Draw the epithelium of the ducts. Now stain the mucous acini lightly, whereas the serous demilunes and the duct should be stained slightly darker to help you understand the difference. Now begin to add the nuclei to all the mucous acini, flattened nucleus basally placed. The lumen should be large and tubular. Add the flattened nucleus of the myoepithelial cells at the periphery and the rounded nuclei in the serous demilune. Now let's add the nuclei to the intercalated duct and to the interlobular duct. Now look at your diagram. What is left? The connective tissue is left. So now begin to draw the connective tissue in between. Add a few capillaries and the connective tissue, collagen fibers, reticular fibers. Draw the nuclei of the capillaries. And there you have the diagram of your mucous salivary gland with serous demilunes with myoepithelial cells. Let's now develop the last part of today's class, which is serous salivary gland. So for that I am going to draw rounded acini, which are my serous acini, with a few ducts in between, and add a few capillaries. Of course, since it's a serous acinus, I am going to draw pyramid shaped cells. The lumen will be narrow in the center. The duct will have simple cuboidal epithelium. I'll now begin to stain my serous acini. I'll stain them quite dark, whereas I'll try and stain the duct a little different to make it show. Now I'll begin to add the nuclei. Rounded nuclei somewhat placed in the middle of the serous acinus. Color the nuclei. Now let's draw the epithelium of the capillaries. What is the cell that we have not drawn yet? Yes, we have not drawn the myoepithelial cell. Let's begin to add the apical zymogen granules first. Now let's add the myoepithelial cell here towards the edge of a serous acinus. Draw its nucleus, flattened and quite dark nucleus. Now let's add the connective tissue element. Let's add the collagen and the reticular fibers and a few nuclei. And there you have the diagram of your serous salivary gland. So this is how, if you know how to draw a serous acinus, a mucous acinus with serous demilunes, myoepithelial cells and a few ducts, it's very easy for you to develop the diagrams of salivary gland. So thank you students for having the patience to watch this, and I'll see you across the screen in my next microscopy video.